Hi, I'm William Huang, and we're going to discuss the board's relevant topics in embryology. Human development is an extremely complex process that is not yet fully understood. Fortunately, you don't have to know a whole lot about the underlying genetics for step 1, but there are a few genes that you should be familiar with. The product of the sonic hedgehog gene is a member of the hedgehog family of cell signaling molecules and serves many roles in development. It is expressed in the zone of polarizing activity in limb buds, where it plays a role in patterning along the anterior-posterior axis, and it is also involved in dorsal-ventral patterning of the neural tube. Sonic hedgehog spreads from the notochord and ventral part of the neural tube, and its concentration decreases towards the dorsal part of the neural tube, so higher concentrations of sonic hedgehog induce cells to develop into more ventral structures. In contrast, the Wnt family of genes is expressed in the apical ectodermal ridge, which is the most distal part of a developing limb. There are many different Wnt genes that can induce different tissue types, and in early embryogenesis, Wnt7a plays a major role in limb development. Both the Wnt family of genes and sonic hedgehog are extracellular signaling molecules that determine cell differentiation and proliferation. Unregulated activation of sonic hedgehog and Wnt genes can also cause tumors. Fibroblast growth factors also have a variety of roles in embryos and adults, but on the step 1, you will most likely see them in the context of limb development. Like the Wnt genes, they are produced at the apical ectodermal ridge, where they stimulate mitosis of underlying mesoderm, providing for lengthening of limbs. Homeobox genes, also known as Hox genes, are most important for their involvement in segmental organization of an embryo in a cranial-caudal direction. As you can see in this image, different combinations of Hox genes cause different segments to have distinct fates. So, for example, Hox D13 is expressed in the metacarpals and phalanges, but not in the carpals. Incorrect expression or mutations in Hox genes can result in major changes in development or localization of appendages. Now let's discuss the general timeline of development. This information can be useful in many cases, such as knowing when teratogens are most dangerous and knowing when congenital defects can be tested. Let's start from the beginning with fertilization. First, in the fallopian tube, a sperm finds a secondary oocyte that has its cell cycle arrested in meiosis II. It undergoes an acrosomal reaction to release enzymes which degrade the zona pellucida of the oocyte, allowing the sperm to pass through so that the gametes can fuse. In response to membrane fusion, the oocyte releases enzymes from its cortical granules, causing the zona pellucida to become impenetrable to additional sperm. The oocyte then finishes meiosis II and releases a polar body. The nuclear membranes of the oocyte and sperm are then degraded and reform around the diploid genome. At this point, it is a single-celled zygote. In week 1, as the zygote travels through the fallopian tube, it is cleaved into progressively smaller daughter cells. By about day 3, the zygote has become a morula, which is a compact ball of about 32 cells, which has reached the uterus. Next, it turns into a blastocyst, in which the cells surround a central cavity called a blastocele. By the 6th day, the embryo implants in the wall of the uterus, and human chorionic gonadotropin, or HCG, secretion begins shortly after. Since pregnancy tests detect HCG, it will not be able to detect a pregnancy until after implantation. Do you know which structure prevents implantation before day 6? The zona pellucida, which is not shed from the embryo until the blastocyst stage. During the first week, the only differentiation that occurs is into the trophoblast, which will become the placenta, and the inner cell mass, which will become the embryo, amnion, and yolk sac. During week 2, the inner cell mass begins to differentiate into the precursors of different tissues. The inner cell mass divides into the bilaminar disc form, which is exactly what it sounds like. A disc composed of an epiblast layer and a hypoblast layer, with the epiblast located dorsally and the hypoblast ventrally. The epiblast is mostly associated with ectoderm. It is actually a precursor to all 3 germ layers, since it invaginates to form the pyrimidine streak, which gives rise to both the mesoderm and part of the endoderm. As you can see in the image here, 2 cavities form as well. The dorsal one is the amniotic cavity, the ventral one is the yolk sac. By the third week, all three tissue layers exist: ectoderm, mesoderm, and endoderm, and the most basic forms of organ systems begin to develop. Gastrulation occurs, creating a tube through the embryo that will eventually become the gastrointestinal system. The notochord and neural plate begin to form, which are necessary for the development of the central nervous system. During week 3, the process of neurulation continues. The notochord induces the ectoderm above it to form the neural plate. The neural plate invaginates, forming the neural groove at the midline and two neural folds laterally. The plate rolls up like a tube with the edges of these neural folds, the neural crests, fusing together at the midline. This neurotube formation begins in the neck region, the fourth somite, proceeds in both cephalic and caudal directions, the cephalic being completed first. Do you remember which vitamin deficiency most commonly causes neurotube defects? If you said folate, you're right. Next, the cephalic end dilates, forming the forebrain, midbrain, and hindbrain, while the rest of the neurotube forms a spinal cord. As the neural folds elevate and join at the midline, cells at the edges of the lateral plates, or neural crests, differentiate to form neural crest cells. When they reach their final destinations, they differentiate into numerous cell types, including spinal ganglia, melanocytes, and support cells of the nervous system. Gut tube formation is concurrently occurring ventrally, forming a tube beneath a tube held together in mesodermal glue. What disease is caused by failure of neural crest cells to migrate to become colonic ganglion cells? Hirschsprung's disease. Failure of neural crest cell migration leads to a colon that cannot relax and is functionally obstructed, with dilation proximal to the aganglionic region. Most organ development occurs during weeks 3 through 8, which makes this the time that the embryo is most susceptible to teratogens. During week 4, the heart begins to beat and limb buds begin to form. At this time, the heart is just a single contracting tube, and it does not have all 4 chambers until week 7. During the 5th week, muscles begin to develop, craniofacial development begins, so that by the beginning of the fetal period, week 8, the fetus is able to move and looks like a baby. By week 10, genitalia have male or female characteristics, so this is the first time that parents can determine the sex of their baby by ultrasound. Lastly, the alar and basal plates are the dorsal and ventral parts of the neural tube, respectively, which are created with the neural tube during the 4th week. The alar plate gives rise to a lot of the sensory parts of the nervous system, and the basal plate contributes to most of the motor components of the nervous system. We already talked about formation of the neural plate and neural tube, but let's quickly cover the role of the notochord. The notochord is like a rod of cells that run along the craniocaudal axis. One of the first roles of the notochord is to induce the formation of the neural plate by secreting the morphogen sonic hedgehog, as we discussed earlier. Later in development, the notochord becomes the nucleus pulposus, which is a jelly-like substance between the intervertebral discs. Let's go over some useful correlations to help review and remember some of the facts we just covered. You can remember what happens during the 2nd week of development by remembering the rule of 2s. There are 2 germ layers, the epiblast and hypoblast, 2 cavities, the amniotic and yolk sac, and 2 components to the placenta, the cytotrophoblast and syncytiotrophoblast. The rule of 3s for the 3rd week is that there are now 3 germ layers, ectoderm, mesoderm, and endoderm. Lastly, the rule of 4s for week 4 is that this is when the 4 limb buds start growing, and this is when the heart starts beating. The 4 chambers of the heart don't finish developing and becoming separate from one another until week 7, but thinking of the adult 4-chambered heart can help you remember that it starts beating during week 4. Knowing the embryonic origins of organs and tissues can be helpful in answering some Step 1 questions, and may also help you understand organ system, anatomy, physiology, and pathology. For example, Rathke's pouch is derived from surface ectoderm and gives rise to the adenohypophysis, but can also develop into a craniopharyngioma. Since this slide is mostly lists of tissue types, I'm not going to read it off to you, but you should definitely come back and review this on your own. While it may look like a lot to memorize, you probably already know a lot of it, such as the fact that the gut tube epithelium is derived from endoderm and the brain and spinal cord are derived from neuroectoderm. So you can focus more on the ones that you don't know. With some exceptions, the types of tissues that derive from the different germ layers often perform vaguely similar functions. So a small amount of pattern memorization will help save you from a larger amount of rote memorization. These terms describe types of errors in organ morphogenesis that are likely to show up on some of your test questions, so make sure you know the differences between them. A malformation can be caused by a variety of things, but its cause is something that occurs within the developing embryo rather than anything outside the uterus. It occurs during the embryonic period, usually weeks 3 through 8, and generally results in a permanent organ disorder. A deformation, on the other hand, occurs after the embryonic period and is due to something outside of the fetus. For example, if there are twins, crowding can cause one twin to not develop properly. Another example of deformation is deformation of the head of a neonate during vaginal delivery. Deformations are generally not permanent and will correct on their own over time. Agenesis is when an organ does not form due to the fact that the primordial tissue that it comes from was never present. Hypoplasia is when an organ does form but is not complete, such as testicular hypoplasia and Klinefelter syndrome. Lastly, in aplasia, the organ also does not form, but in this case, the primordial tissue was present.